So before I start, I want to, this is the first time I'm at NICE, and I'd like to find out a little bit to who I'm speaking. So let's see, who's building hardware here? Who's a biologist? Who's building algorithms? Who's doing theory of computer science? Nice. Truly interdisciplinary group here. I love that. So in the talk that I give today, in about the first third of the talk, something like that, I will talk about things that are directly related to the big scale topics that we've kind of been discussing. And I tried, because I was expecting to be the last person before the discussion, that I should say a few provocative things there. And then afterwards I will talk about a very humble research project that I've been doing together with Eric Jonas. So the talk is titled "At Which Level We Want to Be Neuroinspired." And before I talk about neuroinspiration, I want to give you a caveat, which is, despite the tone with which neuroscientists give their talks, in many, potentially most cases, we really don't quite know how things work. So here's a few examples from my own lab where we failed at showing things that we should have been able to show. For example, in the frontal eye field, it's an eye movement related brain area. It is known from a lot of research using artificial stimuli that it encodes saliency. It tells us where there's something interesting. It turns out that if you use natural scenes, that's not true at all. It is known from, in my field, much of the work that I've been doing over the last 10 years or so, it was about how people move their arms. It is known that generalization happens in intrinsic coordinates. So if I make a mistake, I learn about my own muscles. I don't learn about the external world. That finding is not true. It's known that primary motor cortex neurons do not change their tuning rapidly. It's not true. It was found, it was based on a lot of theoretical and some experimental work. We believed that uncertainty was represented in a relatively simple way by neuron. And in ongoing research in the lab, it looks like it's not true. Now, these things are examples where people were really pretty, at least some of them were areas where people were really quite certain about it. And yet, if one does it carefully and if one wants it to generalize and uses slightly different experiments, it doesn't replicate. What I want to tell you with that is you want to be careful at how you're being neuro-inspired. And just because see, the problem is the following. What we have is we usually have a data set, and then we have an experimentalist that pores over that data set and converts it into a theory that can be represented in 2 sentences. Now, these 2 sentences are then what's the basis of the neuro-inspired theory in lots of cases. And in those cases, it's not quite clear if what you're building based on it is actually going to mean all that much. And I should also say another thing. When we look at this, all the theories that we have, they're really nice often for population averages. And then we look at the data from every single neuron, and there's huge heterogeneity. So some neurons do the opposite of what the theory says, some neurons do something completely different. There's huge heterogeneity, and that's why we want to be really careful how we interpret the findings that we have. Now here comes the main slide where I want to talk about different levels of abstraction. So there's different layers where we can be bio-inspired. For example, we can talk about detailed modeling of spikes. What we just heard is we can say we have a soma, and we have an apical dendrite, and we have local nonlinearities in that, and we can have an arbitrarily detailed model. And there's some principles that go with it. For example, homeostasis. A neuron wants to make sure that it's supplied with all the things that it needs. For example, there must be stabilization of plasticity. If the neuron learns something, it shouldn't immediately lose that. So that's kind of the most detailed level. Then you can say less detailed spiking neurons. We have a point neuron. It's spiking and has some inputs. It has some outputs. There's different principles that start being very attractive, like spike timing dependent plasticity, shunting inhibition. Shunting inhibition you get automatically out of like the whole neuron simulation, and therefore the principle isn't necessary. Whereas once you like reduce it to be a single point neuron, it becomes very important. At rate codes, you can say there's normalization. normalization and lateral inhibition and feed-forward normalization and concepts like that. Many of them you might not need if you have spiking neurons because, say, a refractory period will do some of those things already for you. Then you can say we can abstract even further where we talk about, we don't care about spikes or firing rate. We're just caring about what the neuron says about the stimulus in the outside world. So now basically all the principles that deal with actual spikes become perfectly irrelevant. At that point of time, the only thing that we're interested in is really something about the information that's available there. And we might be interested in, say, sparseness, maybe temporal predictability. We can say those things without relating to how it's actually encoded by spikes. It could be positively, it could be negatively, with some nonlinearity. Doesn't matter, it's the information that we care about it. And I want to talk about one very high level that people don't usually think about is we can go one step further. We cannot just ask, what's the information that one single neuron carries? But we can say, what's the information that a whole population carries? And there the principles might be, say, to which level can the stimuli that I care about be decoded from the entire population? And at that point of time, we can't even say anything anymore about individual neurons. Or, so for example, you can say selectability. If there's a stimulus that I'm interested in, I want to be able to select it out of all of them. It's a version of the binding problem. Bindability. You can also say if you give me a memory system, the brain somehow has a memory system, then you can say a system like a bag of information can be more or less useful depending on how compatible it is with the properties of our memory system. For example, if our memory system has a small number of inputs, then encodings that require many inputs are not as useful as those that encode many. So and I think and that's at some level the reason why I initially wasn't all that happy with Gary's paper, because all those principles, if you look at people writing about principles that can inspire neuro-inspired computation, they are often living at many of these layers at the same time. And I think that by choosing which level we are on leads us to a clearer definition of what the principles are at that level. Great, so there's different levels how you can be neuro-inspired. And, oh, no one even laughs. You can say there's a whole bunch of algorithms that are awesome, that really work, that people are using, and a lot of them are like very weakly neuro-inspired. Yeah, it's like distributed with smooth transition functions, and a lot of like very weak neuro-inspirations. Why is it that weak neuro-inspirations are actually kind of good? It is because we really have two inspirations. We have an inspiration by looking at the brain, but we have another inspiration that comes from trying to understand theoretical computer science. Which algorithms converge rapidly? That's actually totally important. Am I willing to sacrifice a lot of biological realism to make a practical algorithm converge faster? Yes, I am. And so otherwise we'd all be like infinitely Bayesian and so forth. But there's points to be made that we get, our nervous system gets one sample or maybe 100 samples a second. We better quickly find out how we store them, and we can't really afford doing all the possible things we could do with it. So in that sense, I think there's a good point to be made for why you only want to be weakly neuro-inspired. And here's, I want to just give a list of simple inspirations. Hierarchical computation. Yeah, the brain is hierarchical, and kind of a lot of good, well-working systems are hierarchical. And you can say there's another reason why you want to be hierarchical, which is because the world is hierarchical. And all three of them are parallel descriptions. Continuous activation functions, because what they imply about generalization. Translation invariant representation. Yeah, if you look at primary visual cortex and you move a little bit, it looks basically the same. It's translation invariant. And the world is translation invariant, and therefore building that in is great, because building it in is very cheap. We can solve a convolution using Fourier transforms very efficiently, and therefore anything that's based on that makes our algorithms work much faster. Then there's, of course, recurrence and frequency representations. So we might be inspired by, if we look at the human auditory system, hey, the cochlea is doing a Fourier transform, if you want. And therefore, it might be very good if we would deal with signals to also somehow represented like that. So these very weak neuroinspirational cases are very strong. The question is, can we do more? And here's an example of a couple of past failures from my perspective. Everyone has a different way of looking at their models. But if I look at my own history of, say, what I did during my PhD, there's a lot of computational neuroscience that has this flavor. You say, the brain does something. It does function X. Now, we build X into our simulation, and then we can describe the results of our simulation. But it's very hard what to do with it. Because first, usually the systems we construct like that are not really all that useful for computation. If we just build some random finding from the brain into a computational system, it will generally do much worse than if we don't build it in. And the second one is, it's also very hard to know what we learn about the brain. Here we have the simulation. It has X built into it. Now, and here's what it does. It oscillates, or it synchronizes, or here's a computational problem that I can approximate with it. It's very hard what I'm going to say based on that about neuroscience. So give up. Should we give up? In fact, I did. So let me tell you about my own history. So when I was a PhD student, what I did is I simulated networks. And those were good networks. In fact, very similar to what we just heard. I was simulating neurons that had an apical dendrite and basal dendrites, and they had different function, they had nonlinearity, and they had backpropagation of errors, and it was great, and I was very happy about my simulations. But the problem is, it wasn't explaining all that much data. And I also worked with statistics of natural scenes, and there too, back then, there wasn't all that much data to constrain it. So I did give up. What does it mean I give up? I went to movement science, and I started studying how people solve statistical problems. I did virtual reality experiments with it. I quantified uncertainty, and we characterized in great detail how people solve statistical problems. But I'm here, no? So the fact that I'm here means that probably I didn't completely give up, or at least that I'm starting to convince myself that I was wrong at giving up back then. So here's another reason why I should have been given up. So if you look at how the number of simultaneously recorded neurons is growing in neuroscience over time. Yes, it's exponential. Good news. But no, it's kind of extremely slowly exponential. So it's doubling every 7 years. So that's less than a third of the speed of Moore's law. So yes, if you live really long, if you have a distinguished career until age 80, you're going to see the number of simultaneously recorded neurons grow up by factor 100. But seriously, if you give me 10,000 neurons instead of today's 100, or 200, or 400, however many we have, it's not going to lead to dramatically different results. So another reason to despair. But actually, I think there's a couple of recent developments, in large parts also spurred by, there's multiple factors that play into that. One, there's the Brain Initiative, but two, there's like a large community of technology builders that are starting to push the limits of these things. So I think there's a lot of beautiful new developments for recording technology. One of them is fabrication technology. So at the moment, most approaches that I know of work like that. You have a needle that you stick into the brain, you solder a plug to it, you connect the plug to a preamp, you connect that channel to an amplifier, which from there goes into the AD system, and then you can analyze it. So it's basically incredibly difficult to produce. Because, see, in reality, things should be scaling that Moore's law. If we can make electrical devices smaller, we should be able to produce correspondingly more neurons. So if we can build silicon structures at half the size, or at twice the density, we should be expecting that we can build like little needles at twice the density. But it wasn't scaling at all like that. And I think right now there's a nice development that lots of people are taking this fabrication process seriously. And I think that we will see a factor 1,000 within a few years. The second one, there's the idea of molecular ticker tapes, something that I'm personally very much interested in, which would be a molecular way of recording potentially in huge numbers of neurons. And then there's, of course, optical technologies. And there's so many people working on those ideas that I can't even list them. But given the things that I'm seeing, I have little doubt that we will get a factor 1,000 within 5 years or so. And that would make a big difference. I mean, like a really big difference. And because from my perspective, what's missing for us to build better models is that we just have so pathetically little data. And if you have just little data, it forces your models to be trivial. That's the frustrating things. So if you have very little data that you're fitting your models to, your model has to be very simple, because how could it work otherwise? And then here's some more. Oh wow, well, one of the slides one of the pictures just got lost. So here's 3 approaches, for example, that are pushing the limits for anatomy. Electron microscopy, as we beautifully saw in Sebastian's talks, allows reconstructing whole volumes, and they're pushing the limits there. I should mention a technique that I'm very much interested in at the moment, which is X-ray. It turns out that synchrotron-based X-ray has dramatically improved over the last couple of years and is getting into a comparable range to EM in some cases. And then there's Church has developed PhysEq, which is a way of detecting RNA molecules in the volume where they are, which could be used again to solve long-range tracking problems. And I think all 3 technologies promise dramatically more data. Oh yeah, look, here it is. Here's some reconstructions of neurons that we did based on X-ray data. So, and of course, we need to go beyond cortex. Really, all the other areas are crucially important. Just some examples from the movement system. So now you can say this is a great opportunity for computational scientists like me and like a few other people who spoke today, which is, as these huge data sets come, the problem is moving from getting the data. So at the moment, it's all about, it's so hard to get the data, it's so easy to analyze it, because it's small amounts of data. And I think there will be a strong trend towards the other extreme, where we will be sitting on terabytes, petabytes of data, and it's going to be very hard to make sense out of it. And what we need is to take electrophysiological data, anatomical data, and find a way of understanding the structure, and understand the structure in a way that we could build it into our computational algorithms, but also understand the structure so that we can understand it ourselves, that we can tell it to our students. Because if it's just more and more data, it won't lead to any real understanding. And I should add one aside here that for simulations, I think it's nearly impossible to combine data across neurons, across animals. So, and I should put a caveat for that. What I want to say with that is, if I want to be able to simulate a network of interacting neurons, that is the scenario where it's very hard to use data across animals. Because let's say I find out how each neuron is connected to another one, and I have a data set with the dynamics, I will basically lose a lot of the information that I get by getting the additional data set, by having to solve that assignment problem of how each neuron in the one data set relates to the other problem, relates to the other data set. Oh yeah, one more thing. When people give connectome talks, and I really have to take issue with that, there is often made the assumption that if we could only have the wiring diagram of the mouse, we'd be so much further along in understanding how things work. But yet, if you just give me one wiring diagram of the mouse, I mean, everyone in this room, imagine I give you the hard disk. Here's on your desk, has all the connections in the entire brain. What are you going to do with it? It's very difficult to ask that question, to answer that question. And so I think it's not enough to have one connectome. What we need to do is bring the price of producing connectomes and making sense out of them to the level where one can use that much more reasonable. I believe we need multiple connectomes for most questions that are worth asking. For example, it might be before learning versus after learning. It might be in a world with one statistics versus another set of statistics. Tyson Lee's talk, beautiful. How much cooler would it be if we could say, take an animal, have it grow in this world, see how the connectome looks like, and then have an animal grow up in a different world with different statistics and see how the connectome is different, because that way we'd learn how connectomes adapt to the environment in which we're in. And of course, you could do the same game then with healthy versus diseased brains and so on and so forth. So now that gets me to the second part of my talk. And how many minutes do I have? 6 minutes. Oh, well, then this will be a fast part of this talk. So what we want to do is develop an automatic anatomist. What we mean with that is, so the long-term objective or the long-term vision here is that we want to automate anatomy. And there's actually no alternative to that, no? We are getting these huge data sets, and we need to go beyond just tracing which neuron is which, but we need to start making sense out of it. What are the cell types? What is the microcircuitry? How do neurons tend to connect to one another? And I should say that I've done nothing of that, and all of it is Eric Jonas, and it was a great collaboration. So data types, so we take data from something like from a connectome. What do we have is for each neuron, we have the distances, how far are they away from one another, how deep are they, soma depth, where are the synapses, we could add any other morphological criterion, and of course the connectome, which neuron is connected to which other neuron. It's the binary matrix of connections between them. And I should say, as a background, there's a famous model called the stochastic block model, which works very well if there's no distance dependence. And it works very poorly when applied to real data where there's strong distance dependence. And let me talk about distance dependence. So for most connections, say in cortex or in retina, if neurons are close to one another, they're likely to be connected to one another. If they're really far away from one another, they're very unlikely to be connected. And then there's some transition. And that transition might look like a sigmoid like this. So what we're going to do is we're basically going to say, So and then we have, so what we do is we have soma depth, which we model as Gaussian, synapse depth profile, which we model as mixture of Gaussians, and then we produce a statistical model. Now the statistical model is really the intuition behind it, and I only have time for the intuition here. The intuition behind it is very simple. If we have a pair of neurons, every neuron has a neuron type. The probability that two neurons are connected depends on the two types and the distance between them. Some neurons will connect to one another more or less regardless the distance, and some neurons will connect to one another only if they're at the same place, and yet other neuron types might essentially never connect to one another. And then what we do is we have an algorithm, and we do all the usual tests, and given the time, you'll have to trust my word for that. We know that the algorithm is doing the right thing. We understand what the artifacts are. We have ways of dealing with it. We take the mouse retina data set that Sebastian so nicely introduced earlier, and then we apply our algorithm. And what it should tell us is what are the cell types and how does the connectivity between them look like. And here we have the connectivity matrix after resorting by the various cell types. This is the cell type by cell type distance dependent connectivity function for that. And what you see is in red is the fit that our model has to that. In blue you have the real data. And what you see is our model has a reasonable good fit at fitting that. And now this is the important graph. Now what we have is we have a way of taking the neurons, taking basically the connectivity and other features, to decide which neurons should be of the same type. And now this is our attempt at quantifying that. Now there's two things, two measures that we're using. There's the red measure, which is given our model, how good are we at predicting which neurons will make a connection with one another. What we have in blue is cluster accuracy, which is how similar is the clustering by the algorithm to the clustering of the anatomists. Now, it doesn't mean that the anatomists are right. They will be biased in their own way, but at least it's something that we can compare. And now what we have is here, on the left-hand side, we have the distance as indicated by human anatomists. So the clustering according to human anatomists. What you see is it's very high. It's at about 0.9. So humans are actually pretty good at classifying cells. Well, that's a relief. That's what their life is about. So, and now if we take humans, we just take the cell types, we remove the distance dependency, then of course they do quite a bit worse. Now what we can do is we take our neurons with we take just the connectome, and we ignore the distance, and then we are doing worse than humans, but we at least slightly agree with them. And then we can say we take the connections, the connection matrix, and we also give it distance dependence. We basically allow the probability to decay with distance. And then you see that we're doing much better. We're doing almost as good as the humans. And in fact, we can then give to the algorithm more things that humans use. We can give it the depth of the soma, and we can give it the distribution of synapses, which we know that anatomists are using. What's happening in this case is we're getting worse, slightly worse at predicting which synapses are there, and we're getting better at agreeing with humans, which might suggest that humans actually are excessively relying on the anatomy of cells when classifying them. And lastly, there's a well-known statistical model called LPCM, latent position clustering model, which is quite a bit worse than the algorithms that we have. Now, I'll have to skip over the rest. It does meaningful things when we apply to C. elegans, although this is not what our algorithm was for. Our algorithm assumes that we have a sheet where only the distance matters, whereas the C. elegans is this long animal that doesn't have sheet-like structure at all. We can apply it to the connectome of a classical microprocessor, the MOS 6502, and it pulls out reasonable things even here. So last but not least, I want to emphasize that the model that we had, it's a very early first step. Yes, we use distance dependence and connectivity, but there's clearly multiple overlapping types in cortex, and the way we phrase this problem wouldn't allow us to deal with that. There's also continuous factors. Say a neuron has orientation tuning on top of maybe being a layer 5 pyramidal neuron. And there's far more types of data that one can use and that one could build into these algorithms. I should also just mention one sentence. The algorithm that we use is Markov chain Monte Carlo on large clusters, and we're going, of course, through all the ways of making it run fast that many people in this room have dealt with in the past. And that brings me to my take-home message slide. I think we should really ask seriously the question of which level of abstractions we want to be at. And I think, in particular, for this very high level, I don't think we have thought enough about how we could talk not about the information in individual neurons, but the information of a whole group of neurons, or of a whole area maybe, and be inspired by that. And I do think that we will need automatic anatomists, and I hope that the work that we're doing is going to be superseded by much better ways of doing that soon. And what I have shown you is a way of taking connectomics and getting from there to cell types and microcircuitry. Well, with microcircuitry, I mean the distance-dependent, cell type-dependent connectivity. And with that, thank you very much.
So I wanted to suggest that there's an important thing missing in the discussion, and that is even if one had 100 mice connectomes of a particular area, you might be mystified, because if you focus on connectivity probabilities and likelihood of connectivity between particular cell types in the cortex, for example, as you were talking about, you could learn at the end of the day the average probability of connectivity, maybe as it depends on distance. But of course, the wiring self-organized, and you wouldn't be surprised if two stellate cells that ended up being tuned to the same orientation were more likely to be connected to each other than to other stellate cells, and in turn they would be more likely to wire together to a common target than other stellate cells that had uncorrelated physiological response. Because during the wiring process, that activity shaped the wiring that ended up stabilizing. So, the important ingredient is now available. There's been a little bit of work, and I think a lot more will be coming in the coming years under the Microns program, to put in register the firing during sensory processing in 3 dimensions, with then the connectomic reconstruction of that wiring. And then finally, you get really a new era of information that I think ends up causing a rethinking of a lot of the conclusions that you came to. I wondered if you could address that.
Okay, so I'm not sure if it causes rethinking. So what measuring, what you just sketched would allow measuring tuning curves at the same time. And having that is really a necessity, and I fully agree with you that that is an important step. And I think that certainly at the Microns project, I expect that most projects will be doing that, say, doing calcium imaging beforehand. But tuning curves, what do they really do? They add a continuous factor to each cell. Now you can say a cell isn't just a cell type, but the cell is also tuning to orientation and spatial frequency and things like that. And now you can say the probability of a connection will now depend on the two cell types, but also on those latent continuous variables that describe the tuning curve. And characterizing them will make the model much stronger, will make such models much stronger. But I don't think I don't think any of the first 15 slides or so of my talk would be changed if we had that.
Well, I'm not so much trying to tackle any of the particular slides of your talk, just to say that maybe a more affirmative point, that there's really a new era that will be opened up, because then you can look at the wiring that gives rise to higher order feature self-organization. If you look at the correlated firing during sensory processing, the wiring between those cells, their wiring selectively to targets, you really get at the underpinning of the self-organization of coding, higher order features, that's relevant, and that's completely absent if you don't have the conjunction of the sensory activity with the wiring.
Right, and you can say it's actually a first failure point of this general approach. It is possible that we'll fail at that level. So, so let's say you give me the connectome of a neural circuit, and we have the activity recordings from it. Then the assumption that is behind virtually all the connectome projects is that there is a clean relation between the connectome and the dynamics that we're seeing. And it's going to be absolutely beautiful as soon as the data comes in to find out how good that is possible. And it is the first point at which we might find out that we are on the wrong path.
We could. Or in the alternative, we could really open up a new era of understanding cortical circuitry.
And that's the basket that we're all putting our money into, no?